Meiosis and sexual reproduction. Gametes are eggs or ova in females and sperm in males. Gametes contain only one full set of chromosomes, making them haploid. Early in the development of the embryo, specialized diploid cells called germ cells are produced within the gonads, which are the testes in males and ovaries in females. These germ cells are capable of mitosis to perpetuate the germ cell line, as well as being able to undergo meiosis, producing haploid gametes. When gametes fuse, they form a zygote. A zygote is a fertilized egg, and it contains two sets of chromosomes, and therefore are considered diploid. Most animals and plants, and many unicellular organisms, are diploid, and therefore have two sets of chromosomes in all of their somatic cells. Cells of the body that are not gametes are called somatic cells. In each somatic cell of a diploid organism, the nucleus contains two copies of each chromosome, called homologous chromosomes. One set came from the sperm, and the other came from the ovum. Let's look at some differences between the processes of mitosis and meiosis. First, we'll look at mitosis. Mitosis occurs after the zygote is formed, resulting in the growth of the organism. Mitosis is also used to replace old, worn-out somatic cells. In the process of mitosis, two genetically identical daughter cells are formed. In contrast, meiosis occurs in the formation of the gametes, which are eggs or sperm. Meiosis starts with a diploid parent cell and results in 4 haploid gametes. In other words, there is a reduction in chromosome number by 1/2. Gametes are genetically different from the parent cell and from one another. In order to achieve this reduction in chromosome number, meiosis consists of one round of chromosomal replication followed by 2 rounds of nuclear division. Meiosis consists of meiosis I and meiosis II. The phases of meiosis are as follows. Meiosis I includes prophase I, metaphase I, anaphase I, and telophase I. Meiosis II includes prophase II, metaphase II, anaphase II, and telophase II. Meiosis is only a small part of the cell cycle of the germ cells. The cell cycle of the germ cells includes both interphase and meiosis. Most of the cell cycle is interphase. Interphase consists of the G1 phase, the S phase, and the G2 phase. The G1 phase, or first gap phase, is focused on cellular growth. During the S phase, the cell copies the DNA of the chromosomes. And in the G2 phase, or second gap phase, the cell undergoes final preparations for meiosis. During DNA duplication in the S phase, each chromosome is replicated to produce identical copies. These identical copies are called sister chromatids and are held together at the centromere. Before DNA replication, each chromosome appears as a single line. After DNA replication, each chromosome consists of 2 sister chromatids attached at a centromere. These appear as an X. The chromosome number is still the same at this point, even though there is twice the genetic material in each chromosome after DNA replication has occurred. In early prophase I, the nuclear envelope begins to break down. Homologous chromosomes are paired up and held together by a synaptonymal complex. This tight pairing of homologous chromosomes is called synapsis. In synapsis, the genes on the chromatids of homologous chromosomes are aligned exactly with one another. In this configuration, the 2 homologous chromosomes can be referred to as tetrads because they contain 4 sister chromatids. The synaptonymal complex has recombination nodules that are located at certain intervals along its length. These recombination nodules are the locations where crossing over will take place. Crossover, or genetic recombination, is the exchange of genetic material between non-sister chromatids. In late prophase 1, or prometaphase 1, the spindle fiber microtubules connect to the kinetochore proteins at centromeres. These microtubules grow from the microtubule organizing center, or MTOC. The MTOCs are centromeres that are located at opposite poles of the cell. The microtubules from each pole move toward the midline of the cell and attach to one of the kinetochores of the two fused homologous chromosomes. Each member of the homologous pair attaches to a microtubule that extends from opposite poles of the cell, so that in the next phase, the microtubules are able to pull the homologous chromosome pairs apart. At the end of prophase 1, or prometaphase 1, each tetrad is attached to microtubules from both poles, with one homologous chromosome facing each pole. The homologous chromosomes are still held together at the chiasmata. In addition, the nuclear membrane has now completely broken down. In metaphase 1, homologous chromosomes are aligned at the metaphase plate, which is located roughly in the midline of the cell. The homologous chromosomes are arranged with the kinetochores facing opposite poles. Each homologous pair is oriented randomly at the equator. This increases genetic diversity because each gamete will only end up with one homologous chromosome. To summarize, meiosis 1 creates genetically diverse gametes in two ways. First, crossing over, and then independent assortment. During prophase 1, crossover events between non-sister chromatids of each homologous pair of chromosomes generate recombinant chromatids with new combinations of both maternal and paternal genes. Also, the random assortment of tetrads on the metaphase plate produces unique combinations of maternal and paternal chromosomes that will make their way into the gametes. In anaphase 1, the microtubules pull the linked chromosomes apart. The sister chromatids remain tightly bound together at the centromere. The chiasmata are broken in anaphase 1, as the microtubules attached to the fused kinetochores pull the homologous chromosomes apart. In telophase 1, the separated chromosomes arrive at opposite poles. Then cytokinesis occurs, which is the physical separation of the cytoplasmic components into two haploid daughter cells. At the end of meiosis 1, the cells are haploid because there is only one of each pair of the homologous chromosomes. Each chromosome still consists of two sister chromatids. In meiosis 2, these two sister chromatids will separate from one another, creating four haploid daughter cells. The mechanics of meiosis 2 is similar to mitosis, except that each dividing cell has only one set of homologous chromosomes, each with two chromatids. Therefore, each cell has half the number of sister chromatids to separate out as a diploid cell would during mitosis. In prophase 2, new spindles are formed, and microtubules from these spindles attach to the kinetochores. Each sister chromatid forms an individual kinetochore that attaches to microtubules from opposite poles. In metaphase 2, the sister chromatids line up at the equator of the cell. In anaphase 2, sister chromatids are pulled apart by the kinetochore microtubules and are moved toward opposite poles. In telophase 2, the chromosomes arrive at opposite poles. The chromosomes then decondense and the nuclear envelope forms around the chromosomes. Finally, cytokinesis separates the two cells into four unique haploid cells. Thank you for watching.